Okay, we can get started now. So, hi everyone, welcome to this week's Machine Learning for Protein Engineering seminar series. This week we are pleased and lucky to have David Belanger and Neil Thomas with us. Today they will be presenting engineering highly active and diverse nuclease enzymes by combining machine learning and ultra-high throughput screening. Bit of background for the presenters. Neil is a research scientist at EvolutionaryScale, developing AI tools for protein design. He previously worked at Google X, where he designed proteins by combining machine learning with high throughput screening. He completed his PhD in computer science at UC Berkeley, where his research focused on developing and evaluating methods for protein representation learning and especially protein language models. David Belanger is a research scientist at Google DeepMind, using machine learning to help understand the function of natural proteins and to engineer novel proteins with desirable properties. He received his PhD from UMass Amherst on machine learning methods for structured prediction in NLP. David and Neil, please take it away.
Thanks, Tianyu. Yeah, and thank you for having us at this seminar. I've been following this seminar for several years, so it's an honor to finally be able to speak. So yeah, today David and I are going to be presenting some work that we did at Google on engineering enzymes. So I think this, yeah, I hope this stimulates an active discussion, and we'll have some time at the end for questions. The overall roadmap here is we're going to talk about the project goals and the structure of the enzyme engineering campaign. Then we'll talk about the machine learning methods that we use to design our libraries. We'll talk about a high throughput screening technique that we used and our methods for data collection, and then go over some of the results of our campaign, and then have some time for discussion at the end. So first, what did this enzyme engineering campaign look like? So we really set out with the goal of designing a particular enzyme, and over the course of that campaign also learned a lot about the techniques and tools that we were using to do so. But as an overview, for many of you this is very familiar, but as an overview, the stages of an enzyme engineering campaign usually start with target finding, followed by some discovery, optimization, then scale up. So in the target finding stage, we identify some desired characteristics that we want our enzyme to be able to, for example, to operate in a particular temperature or have some certain function. Then we look for an initial target, or an initial enzyme in the discovery phase, usually mining by metagenomic sequences to find some enzyme that has non-zero activity. And then we take that enzyme and do some optimization procedure so that it improves the activity, thermostability, solubility, a host of desired properties in the optimization phase. And then when we want to scale that up and productionize it, we optimize the production conditions, like a fermentation tank, in order to produce a laundry detergent, for example, in really high throughput. So in this seminar, you've had many people come to talk about both the discovery and optimization phases, and there's a ton of really exciting work on the discovery side, including structure condition generation and diffusion models and language models. But in this talk, we're going to do a deep dive on an optimization campaign, and I hope that that will also be very interesting. So what were we trying to optimize? We're trying to optimize an enzyme called nuclease, which is a non-specific endonuclease, which hydrolyzes both single and double-stranded DNA substrates, shown here in this light orange, interacting with the enzyme, which has some active site at its core and the DNA, where the DNA gets cleaved, essentially. And nuclease is isolated from Bacillus licheniformis and operates optimally in pH 9, so in basic conditions. And the reason that we cared about this particular enzyme was to unlock some of its uses as a potential therapeutic. So the target clinical application here would be to degrade biofilms that accumulate on chronic wounds. Biofilms, bacteria that create these chronic wounds, sort of excrete strands of DNA that allow them basically to form a little shield, and that confers onto them some antibiotic resistance. And so the goal of this campaign, we have this non-specific endonuclease we could use to degrade these biofilms, but it operates at this pH 9. And when we change to a therapeutic pH, so the conditions that would be necessary for this to be a therapy, we take this 80% reduction in enzymatic activity, which means that it would no longer be effective. So our goal was to restore and subsequently improve nuclease activity to unlock this capability, potentially use it as a therapeutic. So that was our protein optimization goal. And along the way, we also sort of had this methods research goal of demonstrating that ML-guided protein design can improve over directed evolution when both use extremely high throughput experiments. So this is sort of a result that I think a lot of us believe to be true, but hasn't been shown in this sort of like head-to-head environment where both types of protein optimization techniques get access to the same level of resources, essentially. So how did we get this high throughput screen? So this is one of the, I think, really cool aspects of this project was we got to work with this ultra high throughput screening experimental platform using a microfluidic sorting device. So just to show what this looks like, what the microfluidic sorting device allows us to do is after we construct our library, transform it into cells, encapsulate those variants into droplets, so we get this water in oil droplet emulsion, we can then pass these through a microfluidic device and measure some output, like in this case, fluorescence. So the little glowing dots are cells that are fluorescing. The ones that are not flashing white are not fluorescing, and they're being sorted into sort of the failed pool, and the successful pool is going down to the bottom. And this device can sort thousands of droplets per second, and this allowed us to sort thousands of protein variants. And a technique like this, we expect to improve both the performance of machine learning guided library design as well as directed evolution. So kind of pit them head to head here. And the other thing I wanted to mention is that this cell sorting technique traditionally is used in this sort of iterative improvement of variants that akin to directed evolution, where we sort variants or we sort our libraries at sequentially higher thresholds. So we say, okay, if fluoresce is at some particular level, then we resort the successful library at a higher level and at a higher level. And then the final pool, we take and isolate, purify, and do some tests, for example, in a plate-based assay. And this technique can be used to optimize the performance of enzymes. The other we can take this same microfluidic or ultra high throughput screen and use it to collect data for modeling. So in this case, what we're going to do is sort in parallel at multiple thresholds, then take the outputs of those sorts, run some sort of sequencing, and then compute a score, which I'll get into more detail later on. But that score will then be assigned to each variant, which we can map back using the results of the sequencing. And that gives us a data set where each variant is linked to some score, and we can use that for training models. So I talked about how one of our goals was to show that we can outperform a baseline directed evolution technique. And so we compared to two different directed evolution techniques. One I'm going to call directed evolution. This is just the fully in vitro independent campaign where we do some mutagenesis. So we start with some initial target, introduce some random variation, or recombine things in that library and then screen and continue that iteratively. Another method, which is akin to an in vitro technique, but that we actually designed in silico, is our hit recombination library. So here, this is a model-free way of improving variants round over round, where we take the results of a screen and say if A and B, two variants are both good, then in the next round, we'll just take the mutations from A, add them to the mutations from B, and screen them in the subsequent round. And just wanted to emphasize that these are very successful techniques for protein engineering. I pulled these two tweets from some recent discussion around the ESM-GFP design, but the recombination space is very dense in functional proteins. And as Deborah marks, I liked this quote of chance favors the prepared genome. And these two techniques are quite good at optimizing and also quite good at finding functional sequences. So this is what we're up against. And our optimization, the overall structure of our campaign looked like this, where we did 4 rounds of design. So we'll call them generation 1, 2, 3, and 4. We started with our wild type nuc-B sequence, generated an initial library G1 using error-prone PCR. So this is just introducing random mutations to the wild type, and then ran an independent directed evolution campaign where this is fully in vitro. We used our screen in the way that I was describing, where we introduce random mutations, recombine successful variants, and then put them back into screening for the next round. And then we also did 3 rounds of designs in parallel, where we ordered the libraries explicitly and then screen them using our microfluidic setup. And here we are screening both the machine learning guided designs and the hit recombination designs in parallel across these 4 rounds. And to give a sense of the scale of data here, because we're doing this, because we're mapping the results using sequencing, we could potentially have screened many more variants just using the ultra-high throughput screen on its own. The directed evolution campaign, for example, can screen hundreds of thousands of variants in principle, but in terms of the actual data that we get for training models, this is what this looks like. For training models and assessing the performance using this sort of like a data collection process, we got about 10,000 data points in each round, where in the designed rounds, around 9,000 of them we designed using machine learning, and between 1 and 2,000 we designed using our hit recombination baseline. So there was one more goal that we had. So we both wanted to show that we could outperform directed evolution and hit recombination across a multi-round optimization campaign. But we also wanted to show that we could have generated a better initial library using some sort of zero-shot design technique, and show that, taken together, all of our techniques can be used to use machine learning guided directed evolution to optimize a protein from just the initial target. And here we're comparing ourselves to the error-prone PCR, where we say, okay, can we actually do better at generating an initial library than some random mutagenesis? Okay, I'm going to hand to David for the first part of the method section, which is about our methods for ML library design.
Great, okay. Next slide, please. Okay, so yeah, we have this framework that sort of unifies all the different techniques we used in this project, and we call it TeleProt. The idea being that this is a planned teleport, meaning that we kind of jump far. So jumping far in sequence space is something that is hard for directed evolution to do, particularly if there's like fitness valleys in between, and that's something that is an opportunity for machine learning to do better. So the framework has like 4 key ingredients, and then I'll talk about sort of specific instantiations of this framework that we used. So in our particular project, we considered only substitutions to the sequence, basically just because it gets complicated otherwise. But like, indels are known to have a high impact on fitness and would be interesting to consider in the future. We also pose an acquisition function, which is just like a fancy term for some sort of sequence to number model. One thing to note is that like at no point in the project do we consider protein structures in terms of our actual sequence design. So everything is just in terms of protein sequences. The next idea is candidate generation. So if you have some in silico, surrogate for an expensive experiment, this F function, finding new sequences that have high model score can still be non-trivial because it's a large search base, even in a computer. So there's some details there. And then we have some heuristics for diversifying our designs, which I'll talk about in a second. Okay, so next slide. So first concept, quite straightforward, is that we train some supervised models. So the idea is that we had data from prior rounds of experiments, we fit a convnet on them, did sort of like turn the crank of typical machine learning where, you like divide it into train and test sets, tune some hyperparameters, blah, blah, blah. And the data set here came from this approach that Neil was describing where we screen at multiple levels in parallel. And so it's actually like it's a discrete multi-class classification model because there's like sort of limited resolution to the readout of the assay. Next slide, please. We also have an unsupervised model, a VAE, which we use at various places that I'll describe more in the subsequent slides. And so here, it's just like quite similar to this deep sequence model. It's like a fully connected MLP for both the encoder and the decoder, trained on a multiple sequence alignment. You may be wondering, why do we not use a protein language model? I kind of like these models. Like, I think they're very easy to reason about. They're very lightweight, both in terms of like sort of computational requirements and time and all that. And there's like a certain amount of like hand curation in terms of the MSA that I think is kind of like a nice handle on things. Next slide. So there's this phase called candidate generation, which is like given a model that is like, an approximation of the fitness landscape that we're trying to optimize. How do we find things with high acquisition function score? So there are like a couple key ideas here. So one is that we are going to define some sort of trust region, which is basically an area where we trust that the model is reliable. And we're going to constrain our local search to that trust region. We also initialize the local search at places that are sort of known to have good score. And that's just like a way to, enhance the optimization by starting at a good place. And we also use like an ensemble of different local search strategies. The idea being that they sort of define different exploration types of the fitness landscape and can lead to like a more diverse set of candidates. So that's candidate generation number one. Next slide, please. So the alternative thing that we do is we sample from a proposal distribution, and then we select samples that have high model score. And so the key idea here is that the proposal distribution allows us to introduce some external prior knowledge about where we think, well, there's two ideas of prior knowledge here. One is where we think the model is going to be reliable, and the other is where we expect sequences to be functional, kind of like a priori, independent of the experimental data. So you can imagine that like this VAE that was trained on a bunch of homologs has a bunch of ideas about epistasis, which might not be evident in the experimental data in earlier rounds, but can kind of, give us hints as to how to propose variants with lots of mutations that are functional. The other idea is to use, so there's this very popular technique in sort of like traditional protein engineering called ProSAR. It's a way to combine mutations that have been identified as having like positive effect. And we use that as a proposal distribution as well. And so here, the proposal distribution and the trust region are sort of like, similar concepts that we're constraining the search in some way. Next slide. Okay, so then the fun part is that if you just do what we have described so far and you just fit a model and you find a bunch of sequences with high model score, it's really boring. The variants that come out are all like really similar to each other, or there's certain sequences, certain mutations that get just used and used and used. And that's because like, if there's mutations that have high experimental like score in the experimental data and we fit a model on them, of course they're going to have high score. Like that's actually like a good thing to have in some sense. And so what we do is we have like a bunch of sort of heuristics here for diversification of the library. What we do is we define some sort of what we call an extrapolation score on the left. So the idea is that for every candidate identified in the previous phase, we label it with sort of how far from the training data it is, or like essentially how much do we trust models' predictions on it, or like how bold is it to go to that place in sequence space. A very simple way to do this is just to compute its like Hamming distance from things that are known hits in the training data. So these like blue radii coming out from the orange are denoting like gradients in the extrapolation score space. Then the next thing we do is we just hand code some target distribution over extrapolation scores. And this is because like, experiments are expensive. We don't get to do that many rounds of experiments. And so we need to very carefully trade off exploration and exploitation. And this felt to us like a very interpretable way to do it, is kind of reduce the concept of extrapolation to a single scalar and then just like engineer some distribution over that scalar that we want to adopt. And then we have some sort of heuristic greedy algorithm for finding a set of sequences that achieve this target distribution over extrapolation scores and have high acquisition function score. So that, allows us to kind of trade off scores from the model with sort of like prior belief about how bold to be in our exploration. All right, next slide. Okay, so next thing we did is from this VAE. So like a VAE is a generative model over sequences. Like you can just naturally sample from it. That's totally fine. Except that if you train it on an MSA that is really diverse, you're going to get really diverse samples back. And here we don't really want to generate things at the level of natural diversity. We want to generate, like neighbors of a very specific sequence. So we want to generate like variants, not members of a particular protein family. And so we adopted this technique introduced in this paper a couple of years ago from Moderna, where they use this particular procedure for, they basically encode the wild type, which gives you a vector, then or like a posterior distribution over vectors, and then you decode those vectors, and that gives you samples that are like neighbors, where neighborhood structure is defined sort of implicitly by way of the neighborhood structure in the latent space. And of course, there's some heuristics we have to bolt on top in terms of like doing some rejection sampling for things that are poorly behaved. But we found this to be like a very attractive way to sample neighbors. It's fast. It's like easy to manipulate, and I recommend using it. Next slide. Okay, so wrapping up. So what we did is we have this framework where we have these different ingredients. There's like acquisition function, candidate generation, and batch selection. Batch selection was basically the same for all approaches, and we just mixed and matched acquisition function approaches with candidate generation approaches. Broadly speaking, the idea here was that we wanted to transition over the course of the campaign from relying on evolutionary data to relying purely on the experimental data at hand. So you can imagine that like as data accumulates, you like trust your acquisition function more, and you can just like kind of brute force optimize it. Whereas in earlier rounds, you need to have like more priors baked in terms of like a candidate generation strategy that uses like a proposal distribution. And I encourage you to refer to the paper for more details about these like particular instances of the framework. Next slide.
Maybe it's just worth, because we're going to present a bunch of results on MBO-DNN. David, do you want to just say one?
Yeah. Okay. Great. So yeah. So the bulk of the results that Neil is going to present on are from the fourth round, the final round of experiments, because that's like the best stuff came from the final round. And the technique that we use there is this thing MBO-DNN, model-based optimization with a deep neural network, which is like kind of what I would say is like almost like the most vanilla version of this, where it's like you have a deep neural network acquisition function, and you do some sort of randomized local search without any sort of explicit priors from evolution anywhere in the stack. But that's like sort of possible because of the fact that a lot of data has accumulated from earlier rounds.
Cool. I'm gonna dive into a little bit more detail on the way that we actually collected training data for our models that incorporated experimental data. So I'll cover the key ideas here. So the first key idea is what's called an enrichment factor. So I'll just walk through a very simple example. So the way that our screening framework works is that we start with some initial library. That library in this toy example has two different variants, gray and green. We'll call them variant A and B. And if we ran some, let's say, if we ran some sequencing experiment with coverage 1, maybe we would get 5 counts that map to variant A, 5 counts that map to variant B, and we would say each of them make up 50% of our initial library. After we pass them through our sorting procedure, we'll have some set of variants that passed our threshold, so here represented by this line here, and then we can sequence we can run sequencing again on this post-sort pool, and what we'll see is that all of our variants A dropped out because they're not glowing. Variant B passed through our filter and became 100% abundant in our post-sort pool. So the abundance of variant A was now 0%, after sorting and 100% for variant B after sorting. So we can compute the enrichment factors here, which is just the ratio of the post-sort abundance to the pre-sort abundance. And so variant B would, in this example, receive an enrichment factor of 2, and variant A would receive an enrichment factor of 0. And this is the way that we can run this procedure with a much more diverse initial library, or with many more variants, and assign enrichment factors, scores to the entire library. So the second key idea that we used to get this sort of multi-class label that David was mentioning is to use fiducial sequences to calibrate our hit calling procedure. So what I mean by a fiducial sequence is just a variant that has some known activity. So for example, we might have some negative controls that we know have no enzymatic activity, or our wild type, which we know has wild type activity at the target pH. The next thing we can do is create replicates of those fiducials using synonymous sequences with synonymous codons. And this gives us basically some null distribution over the enrichment factors of a variant with known activity. So the synonymous codons allow us to say, well, the variation in enrichment factor for this particular variant comes both from some sources of technical variation in terms of sequencing error, in terms of sort of the fidelity of our sort, and also some variation due to the differences in expression due to different codons. But we sort of like wrap all of those in. Now, what we get after we have this distribution over enrichment factors is we can, for a new variant, for a new enrichment factor, do a comparison of that enrichment factor to the null distribution using a right-sided t-test. And that gives us a way to assign a p-value to each variant. Say, oh, well, this variant was much more enriched in this sort than some negative control, and therefore we expect it to be a hit. We're also running this statistical test across an entire library with thousands of variants, like I was describing earlier. So we also do some false discovery rate correction and only call variants hits if their p-value is significant after false discovery rate correction. So the last piece here is that we can run sorts at multiple thresholds to give us some data with what I'll call intermediate activity level resolution. So we could in principle get like a quantitative score per variant using these enrichment factors, but we found this to be a nice medium where we were more certain of the signal in our data, and also were able to leverage the experiment to tell us not only is something a binary label of is it better or worse than a non-functional variant, the wild type, some improved variant, but we get all of those labels, and then we can resolve those labels to construct the final data set where each variant is assigned sort of like the highest score, highest consensus score across these three different thresholding experiments. This is not only the way that, just to clarify, this isn't only the way that we get data for machine learning, but it's also the way that we assess the quality of the designed library. So we'll dive into that in our results section. So just as a reminder, so now we've gone over sort of the methodology that we had and also our experimental platform. So as a reminder, our campaign had two different baseline directed evolution techniques, this sort of in vitro campaign, as well as this in silico hit recombination campaign that we screened in parallel. And we did 4 rounds of design. And so when I talk about results, I'll talk about results from G3 and G4. So the first thing we wanted to assess was, did we find something that we could take, follow up and potentially make a therapeutic out of? So here we want to assess the performance of the top variant. So this is a very common way of assessing the success of a protein engineering campaign. So here we'll use our screen to sort at sequentially higher thresholds and then purify the top performers and test them. And our full hit validation funnel goes from this very high throughput, low resolution assay where we're sorting tens of thousands of variants to isolating the top performers, taking them to some plate assay where we assess their performance over time, and are able to get a better measurement of the enzymatic activity. We then take those further for specific activity assessment. So here we're purifying the enzyme and assessing its activity at multiple concentrations, And then the top performers from that proceed to biofilm disruption, where we actually put the purified enzyme in contact with a biofilm and then look to see how it degrades. So sort of our top line result is we found a better variant than this directed evolution campaign. And here I've indicated that we are showing the results after 3 rounds. So after 3 rounds, we actually ended the directed evolution campaign because we had a much better library from the designed library. So here, the top directed evolution variant is indicated by DE3, and it achieved a 12-fold improvement over the wild type in nuclease activity compared to the ML3, or the top machine learning variant, after 3 rounds of design, which achieved a 19-fold improvement. And this is in this specific activity assay. Then just to validate, we took this top variant and put it in contact with a biofilm. And what we see is that the machine learning design variant indeed degrades the biofilm at low concentrations, much lower concentrations compared to the wild type enzyme when we look at this dose response curve. So just one thing to note, I'll talk about, or I've already mentioned this A73R variant, but this is an 8-fold improved variant that was found in the very first error-prone PCR library, and we used as a fiducial going forward. So we can also assess this overall composition of the libraries to say, like, were we more successful at finding hits? Were we able to also create a diverse library of hits? So here, what I'm showing is the proportion of designs that were found to have activity greater than 8-fold improved over the wild type after 4 rounds of designs, comparing the in silico hit recombination campaign to one of our TeleProt techniques. So here, this is MBO-DNN, trained on all the data from 3 rounds and then designing into the 4th round. So here we see that there's a much higher hit rate. And in addition, that hit rate is maintained even when designing far out in sequence space, out to 13, 14, et cetera, mutations. We also wanted to make sure that these machine learning, that our machine learning designed libraries weren't just basically finding some local neighborhood around a successful variant in order to achieve this high hit rate. So we also clustered the successful designs. And what we're showing here is that if we look at just the raw number of sequences designed by each library in the final round, it's about similar, so it's around 1,000 for each of these for the 2 different techniques. But if we then cluster those libraries, cluster only the hits from those libraries, we see that this orange line is far above the green line, where at every cluster diameter, so we say, okay, take all of you don't get credit for a hit if they're only 2 mutations away from each other, only 5 mutations away from each other, et cetera. So the orange line is far above the green line, indicating that our designs in this machine learning guided library are much more diverse than the hit recombination library, which suffers from a diversity problem because it can only recombine things. We also wanted to make sure that that diversity was present in terms of the structural components of the protein. So here I'm plotting for the hits in each library, extracting out the mutations present in those hits and plotting them on the protein structure here. And of course, machine learning has many more hits, so there are many more mutations present. But what's interesting is that these hits, or the mutations present in those hits, span a variety of functional domains across the protein. So all different sheets, different helices, and also across the DNA interface domains, or the portions of the molecule that interface with DNA. And we were particularly interested in a hypothesis that the DNA interface, that the designs increased the positive charge of the DNA interface, which would increase the favorability of interacting with negatively charged DNA. And so we do kind of see that, where, like, the wild type gray dot, all of the hits are a little bit above in terms of isoelectric point. Yeah, David, do you want to talk about the 0-shot initial library design results?
Sure. Yeah. Next slide, please. So just as a reminder, this is like an independently run thing that's like simulating had we done a different initial library. And, the gold standard for evaluating this 0-shot library would be to sort of take it forward with further machine learning guided designs, like basically redo the entire campaign. This is a different starting point, but that's not what we did. That would be too complex. And so instead what we do is we evaluate the zero-shot library against the error-prone PCR in terms of metrics that we think sort of summarize how useful it would be for subsequent optimization campaigns. All right. So it's also worth noting that we actually did this investigation later on. Like we did this in the 4th round of experiment. We multiplexed this with the 4th round of experiments. And the design methods didn't use data from previous rounds, but we had observed sort of retrospectively that models were pretty good at ranking the error-prone PCR variants in terms of functional versus non-functional. So on the left here, we just show that the so we have this zero-shot model score, which is the likelihood of a variant under that VAE that I described earlier. And we can see that there's some separation between these two distributions. And so you can imagine that would suggest that if we select variants with high zero-shot model score, we're more likely to have an actual functional variant. And one way to quantify this on the right is this idea of if we are to sweep over sort of subsampling rates of our library, but we subsample not by randomly subsampling, but by taking the top K using the model score as a way to sort, what fraction of the hits will we actually keep? And if we look at say the middle of the middle x-axis of this figure, so we would keep about 75% of the functional variants if we reduced the library size by 50%. So that's significantly better than the chance, which is the gray line. Okay, next slide. So inspired by the stuff on the previous slide, we said, okay, let's design a new library. And the key difference here is that we're gonna rather than the error-prone PCR library had a relatively small number of mutations per variant because it'd be a massive waste of experimental resources to synthesize or to create error-prone PCR variants with lots of mutations because they're just likely dead. But we said, how far can we push this? So if we sample from this model, can we get designs with lots of mutations that are still functional? So in this slide, the bar for a variant being a hit is relatively low. It's just does it sort of present non-zero enzymatic activity? And we can see that we have a better hit rate and our designs are diverse. So this is great. It's a significantly higher hit rate for this zero-shot sampling than error-prone PCR. And then on the next slide is what we were most excited about, which is that actually we were able to design variants that were by chance better than the wild type. So you can imagine that if you have such a higher rate in the previous slide of finding things that are functional, you're just sort of by the chain rule of probability, more likely to find things that are better as well, because you just have more shots on goal. And so this was definitely borne out in the results where we had actually a pretty decent hit rate, around 10% or so of finding things that have activity greater than the wild type. And here, again, we have relatively strong diversity of these designs. Okay, next slide, please. So we have this big data set and it's open source. So next slide, please. And so I just want to quickly summarize this data set and use this as a call to arms to use it for model development, but also for studying nuc-B as an enzyme. This is a way to probe the fitness landscape. And so this is a pretty different data set than some of the existing open source enzyme data sets in that there's variants with a pretty wide range of numbers of mutations from some wild type sequence. The cost of course is that they're not randomly sampled. They came from the trajectory of some machine learning guided optimization. So there's sort of some path they're taking through sequence space. But that said, if we look on the right, we have the distribution of activity labels from the initial error-prone PCR data. And this data is just kind of challenging to work with if you want to develop machine learning techniques, because there are so few things with activity better than the wild type that it's really hard to assess retrospectively if a model would be able to sort of identify, discriminate between things being better than the wild type or not. Whereas our data, because of course it came from a campaign that successfully improved the enzyme has like a lot more stuff, like 19% versus 1% of things that are actually better than the wild type, and so we can use that to sort of like develop new models. It's also worth pointing out that we like the source of truth we should use for enzyme activity is discrete. Like we have bucketed things into like 4 or 5 discrete activity levels, just due to kind of the resolution of the assay. This is a little bit different than other setups where people look at like Spearman correlation, things like that. Next slide, please. All right, so we have plenty of time, which is great, and I'm hoping that we can like use it to have some discussions from the audience. So Neil and I have like some discussion topics that we can put up on the screen, but like of course we'd love it if other people suggested other things. One thing to point out is that there's like a ton of potential future work, and so like we didn't use protein language models here. Like of course there's a lot of opportunities to use protein language models for zero-shot design, for sort of fine-tuning regression models, and our data set could be a good place to kind of benchmark those models. We were excited about the fact that we didn't use protein structures because it was kind of like simple and straightforward, but like of course there's a lot of opportunities there. The other thing to keep in mind is that we use fairly expensive DNA synthesis approaches. Like we explicitly synthesize libraries of the size like 10,000 variants. It's not clear if that's necessary, like especially in the early rounds of experiments. I can imagine some sort of hybrid approach where you use some randomized DNA synthesis protocol, which is like significantly cheaper, significantly faster, and then maybe in later rounds you use more sophisticated explicit design. And of course, all models are wrong, and like reasoning about the uncertainty of models could be useful. There's also like better methods for processing count data from these NGS-based assays is also like a great opportunity here. Like there's definitely situations where you lose data by only focusing on things where you have high confidence in your assessment of enrichment factors. But if you can kind of drop that down, you could recover a lot more data.
Maybe I'll just summarize here what our findings are, and then we'll open it up to questions. So yeah, I think we were quite pleased with the results of the machine learning guided libraries. I think outperforming directed evolution in the context of a high throughput screen was, it was surprising to see. And so we were very pleased that the top variant that we found at the end of our campaign was indeed seen to be better in terms of specific activity and validated in biofilm assessment. I think the other summary here is that the sort of TeleProt framework that we were presenting that is a good way to balance evolutionary and assay labeled data when designing libraries. So there are a variety of techniques that allow one to incorporate evolutionary or assay labeled data depending on what is available and depending on what is successful. Another finding here, our zero-shot design library was maybe like one of our most successful, like compared to error-prone PCR. This hit rate is kind of outrageous. And this was also quite surprising to see that we could design way farther in sequence space without taking this like exponential hit rate. So, and this was just using an MSA. So we didn't use structure, we didn't use large scale pre-training. So when MSAs are available, I think they are quite powerful for zero-shot design. And then the last point is that the high throughput experiments allowed us to employ a diverse portfolio of sequence design approaches. So we could experiment with all of those different combinations we talked about when we presented that table of TeleProt techniques. And not all of those techniques were successful, or some were much more successful than others. And I think that was a really important part of this study, was not only could we get some statistical confidence in the assessment of the hit rate of each library, we could employ a diverse pool of different techniques, and we could screen in parallel a very strong baseline, which I think gave us a lot of confidence that our techniques were actually working. So with that, I want to acknowledge this was like a huge collaborative effort, especially we got to have this great collaboration with Triple Bar, and also a number of people, both at Google X and now at Google DeepMind. So yeah, this was a really fun project to work on. And with that, I'll open it up to questions.
Okay, awesome. Thanks so much, Neil and David. Feel free to leave questions in the chat and I can read them out, or I can also, you can also unmute as well.
I have one question. Is there a reason why you decided not to use the structure? Because I'm looking at your mutations and they're all like clustered in the structure, like in the, it seems to be like the DNA binding domain.
Yeah, I think that, well, I think, so first, I do think that the mutations were across a wide variety of different domains, but I think in the DNA interface was particularly interesting because it kind of like makes sense from a functional perspective. But I do think that one of the hypotheses we were trying to test actually was we wanted to develop a technique that you could proceed without structure because many enzyme engineering campaigns start with no structure. And so I think that was kind of the goal of the approach, was to show that we could do this without a starting structure. Although it would be really, although like there is a PDB available for this particular wild type. I think one other thing that is important to know is that at least for us, like rational design that's conditioned on structure actually limits the set of hypotheses that you try to test. I think that the sort of success of directed evolution has shown that like mutations appear all over the molecule, and like, you know, you're right that there are a lot in the DNA interface, but another part that's really interesting is there are a lot that just like are elsewhere that don't have an obvious structural interpretation. And so I think that we wanted explicitly to sort of increase the success of our campaign by not limiting ourselves in terms of where to generate mutations on the molecule just because they had some structural interpretation. That's a good question. That's a really great question, though.
Great. Meg had a question in the chat. I guess, Meg, if you want to, do you want to read that out, or I can read it as well. It concerns mostly about multi-objective optimization. So currently the assay is set up for measuring or aiming towards better activity and a more neutral pH for nucleases. If someone wanted to adopt this modeling and campaign regime for their own proteins, do you see TeleProt or TeleProt having the ability to address multiple biophysical properties like stability and substrate specificity in a multi-active fashion? And would discrimination of activity above wild type be too complicated for this simultaneous optimization?
I can take this quickly. So I think that, you know, the natural way to do this would be you just reduce these two objectives to one scalar, right? Like you just like have two measurements and you measure them both. You have a model for both. You have one scalar, and then you do the same kind of, like you have an acquisition function, and then you just kind of turn the crank for what we did previously. Another way to do this is to sort of decouple the problem where we had this idea of like a proposal distribution, which is imbuing some like prior knowledge about something. And it might be that these two objectives, one of them, which is like hard to model, or where we're maybe not even collecting any experimental data for, but we have a lot of priors about from like some external data sources or something. And so there you can imagine sampling from some model that sort of like enriches for things that have high score under that objective, but then screening and modeling the other objective, something like that.
Great, thanks. Next question. Were you able to quantify how much better the VAE was than ProSAR?
Yes, actually, well, okay. We have an experiment where we took ProSAR and we collected real experimental data. We designed variants using ProSAR. And then we had a variant of that where we sampled things using ProSAR, but then rejected things with low VAE score. And that consistently did better. So that's one answer. There's like this like hybrid ProSAR plus VAE, ProSAR plus screen, we called it in the paper. So you should check that out. It was like really successful in round 3 of the experiments, for example. In terms of just like a head-to-head sort of like VAE zero-shot design versus ProSAR, like they didn't happen at the same time. Like one is like a supervised design thing that happened in like ML2, whereas one was like in the zero-shot round. So we don't have that kind of head-to-head comparison.
Great, thanks. Next question. Apart from literature, what went into choosing the model architecture? I think something like a diffusion model to learn the denoising that directed evolution does could work, but maybe want your thoughts on this.
Yeah, I mean, David,
Go for it.
I think that we had seen a lot of success with these two types of models, like the sort of CNN trained on assay labeled data showed a lot of success in some previous work, especially on like AAV design, and this like deep sequence VAE we saw to have success in this like variant effect prediction type of problem. There's also a few other papers that yeah, so like that, like you said, your question was besides literature. I think that was sort of the main motivation there. I also think that we did a lot of this before diffusion models existed, right, David?
Yeah, totally. I think, taking a step back, like there's a lot of moving parts in this project. And I think a lot of the technical complexity was in going from NGS data to like a data set that we can sort of reliably model, and then like reasoning about where we trust that model and where we don't. Like there's this other axis of like sort of sophisticated models on top of that, but the data stack was like kind of complex here.
I think controllability is also an interesting aspect here. Like I think being able to design things that are not so far away in sequence space was another like design decision. Not to say that that same type of capability can't be designed into sort of more modern sequence design approaches, but the sort of like local search as opposed to doing some sort of diffusion and then inverse folding or something, or like learning some edit model. Like I think the controllability in terms of being able to stay within a trust region where like, well, we collected data out to, 4 mutations away from wild type, so we shouldn't probably try to design too far away from that. That was another like design decision, and the models that we chose were amenable to that.
Great, thanks. Next question. How deep was your MSA? How large of an MSA do you think you would need for the zero-shot prediction to work?
Well, first of all, everything's online, which is like, I will answer your question, but I want to just point out that like a lot of the data, all the data that we use in the project is online, and every single figure and table in the paper is produced by notebooks that are on GitHub. I think the MSA was like what, somewhere like 5,000 rows, 3,000, yeah, something like that.
I mean, one thing to note is that like what David was saying before, where like we had this very, very deep, diverse MSA, but then we actually didn't want to sample from exactly that distribution. So I think it is a little bit hard to know how deep that MSA needs to be in order to successfully design variants using this sort of like decoding from the wild type posterior. The one thing I will say is that we have like a way to assess that, which is like you can, not to say that every campaign will have an error-prone PCR library associated with it, but this was kind of our validation that our MSA was good enough, right? Like we found that screening using a model trained on that MSA successfully was able to like screen for functional variants. So that gave us some confidence in that. So that's one way of like assessing how deep the MSA needs to be.
Great, thanks. Next question is, do you think something like this can be applied to enzymes that are harder to assay at scale, i.e. no fluorescent readout, or with very diverse, naturally occurring homologs, such as polyketide synthesis or non-ribosomal peptide synthesis?
Wait, sorry, is the question about the platform or the design technique?
I think about TeleProt.
Oh, okay.
Yeah, I mean, I think that, so there's, roughly speaking, we're relying on 2 sources of data, right? There's like evolution and there's data from an assay. And you sort of like, if you have less of one, you need more of the other and vice versa. And so I think that like, you give those 2 examples and they're like on opposite ends of the spectrum. I think that, better models could certainly help bring the data requirements down. Like I'm confident that with, all these bright grad students out there, we can have some, new zero shot or like few shot learning technique that, you give it 96 variants in their activity and that can have like a great regression model from that. Right. And so I think that, maybe it's just an appeal to more sophisticated machine learning will help us kind of break through that barrier. Everything, of course, becomes easier when you just have a lot of data. That's why we were able to use like a convent that with no prior knowledge at all. It was just like trained from scratch. And then in terms of sort of like the natural diversity where like, you don't have a lot of like homology or whatever, I think either you can rely on protein language models that are trained on like huge, broad amounts of proteins. And there's like some like bridging things that are like, remote homologs or like structural homologs, but not sequence homologs. Or, you just figure out a way to measure at scale and use a sort of simple model like we did.
Okay, great. Another final question in the chat. Could something like this be applied to study fusion proteins? How could you approach generating an MSA for such systems for zero-shot prediction?
Yeah, I think this gets into territory. Well, you could produce an MSA for each component of the fusion separately and then try to incorporate that into your final like acquisition function. But I do think that this is where I would probably appeal to like more modern, like for example, protein language models that are weakly supervised on lots of protein-protein interactions that might be able to give you some sort of score that says, oh, well, this fusion seems more reasonable than this fusion. But like you said, sort of this is pushing at the edges of where the MSAs break down, and you might want to appeal to some model that has learned sort of the distribution over protein sequences just from the evolutionary diversity. I don't know, David, do you have something to add to that?
Yeah, I agree. I think there's a lot of interesting new models out there.
Great. That's all the questions in the chat. Any more questions from the audience?
And feel free to email us also, or put issues on GitHub or whatever.
In particular, use our data. And if you use our data, don't hesitate to reach out if you have questions about it or you're trying to figure out how to incorporate it into your like model development workflow or whatever. We'd love to talk to you about this data set.
Or if you find something with it.
Okay, awesome. Thanks so much, Neo and David, for giving that great talk. And we'll be back next week for another seminar. So thanks, everyone.
Yeah, thanks for the great questions and thanks for organizing.
All right, see you.
Yeah.